Welcome to the third lecture on antimicrobial chemotherapy. In our today's talk, we will be discussing about protein biosynthesis inhibition. Here we will be discussing about those antimicrobial drugs that specifically target the prokaryotic ribosome, that is 70S. Our ribosome is of 80S and hence this leads to the selective toxicity of protein biosynthesis inhibitors. These drugs are having good chemotherapeutic index, but still they have lesser chemotherapeutic index as compared to cell wall biosynthesis inhibitors. If we look specifically the target sites which are being utilized by protein biosynthesis inhibitors, we will see that it mainly comprises of four important steps. Step 1 that is being inhibited is the binding of peptidyl tRNA onto the ribosomes. Second step that is being inhibited is the reading of the mRNA. Third step that is being inhibited is the joining of the two amino acids, or the peptide bonds formation steps are inhibited. Fourth step that is inhibited is the translocation, that is the movement of the ribosomes during protein biosynthesis. Let's start with aminoglycosides. Aminoglycosides are a group of bactericidal agents which comprises of amino acid attached to a carbohydrate moiety with the help of a glycosidic linkage. These drugs were first isolated by Selman Waksman in the year 1943, where he isolated it from Streptomyces graecosus. Later on, many derivatives of streptomycin were isolated, namely kanamycin, nettlomycin, then we have amikacin, then we have gentamicin, tobramycin, and the list is endless. Initially, when the aminoglycosides were discovered, it was seen that how they inhibit the protein biosynthesis pathway. It was found that it binds to the 30S ribosomal subunit. And by binding to the 30S ribosomal subunit, what it does, it actually inhibits the proofreading activity which is being executed during the protein biosynthesis, as a result of which it leads to the misreading of the mRNA. If misreading of the mRNA takes place, then what happens? That faulty amino acids are getting incorporated. This leads to mutations in the protein, and many a times what happens that premature termination of protein biosynthesis takes place. This is one of the ways that it can prevent protein biosynthesis. Secondly, what it does, it has been seen that it prevents the translocation of the ribosome. It restricts the translocation of the ribosome, and in certain cases it has also been seen that it disrupts the membranes. Let's come to the spectrum of activity of streptomycin. They got immense fame because of their ability to combat mycobacterial infections. Later on, it was seen that they show various side effects like damages in the kidney, ear infections, and newer drugs were also emerging with lesser side effects like second and third generation chloramphenical. This actually outshaded their use. Nowadays, we have seen that they are used in combinatorial therapy, as they are used in combination with tetracyclines to treat plague and tularemia. We have also seen that they are used in combinations in treating those patients wherein we see the emergence of penicillin resistance. Aminoglycosides and their various derivatives like amikacin, which is popularly used to treat nosocomial infections or hospital-borne infections. Kanamycin is also used for treating gram-negative bacteria, and we have seen their use in treating urinary tract infections, and tobramycin is used to treat pseudomonas caused respiratory diseases in patients with cystic fibrosis. Means immunocompromised patients are having certain diseases which are being treated with amikacin. Chloramphenical is a broad spectrum antibiotic which is capable of executing its inhibitory action on gram-positive bacteria, gram-negative bacteria, Rickettsia and Chlamydia. How they execute their action? They bind to the 23S ribosomal subunit and prevent the peptidyl transferase activity or peptidyl transferase enzyme is being inhibited. In this way, they prevent the protein synthesis pathway. We have seen that chloramphenical has got wide range of action, but it has got many drawbacks too, as we will be seeing that it causes aplastic anemia, means it prevents the incorporation of hemoglobin into the red blood cells. It decreases the white blood cell count, and as a result of which it has got many more disadvantages, as we will be seeing that it is responsible for causing Gray syndrome. Chloramphenical has got a very good absorption capacity. It's a very small molecule, as a result of which it gets absorbed into the all tissue fluids, and it is responsible for treating meningitis. Tetracycline is a compound having a naphthalene ring, and because of attachment of various derivatives or various side groups, we get various derivatives of tetracycline like chlorotetracycline, doxytetracycline and many more. They are also a broad spectrum antibiotics. They can get readily absorbed into the tissue fluids, but they cannot cross the blood-brain barrier, as chloramphenical can do, but tetracyclines are unable to do so. They have got their action by binding to the 30S ribosomal subunit, and by binding to the 30S ribosomal subunit, they prevent the binding of amino-acyl tRNA onto the A site. Tetracycline has got various side effects, as they destroy the intestinal microflora and thereby they promote the growth of Candida albicans. Secondly, their excessive use may damage kidneys and livers. Their use also have got a detrimental effect on deposition of brown color on the teeth. So we have seen that they have got various side effects, but still they are considered as a two-edged sword because of their ability to combat a wide range of organisms, starting from gram-positive to gram-negative, Rickettsia and Chlamydia. Macrolides are also broad-spectrum bacteriostatic agents. It comprises of a lactone ring and sugar molecules. The most eminent member of this group is erythromycin. This drug is considered to be the drug with least side effects. They are mostly applicable in case of gram-positive bacteria. They have been utilized or working effectively against penicillin-resistant strains of bacteria. They have been used for the treatment of diseases wherein antibiotics like penicillin is ineffective. It is isolated from the organism called Streptomyces erythraeus. And many more derivatives of erythromycin are called azithromycin and clarithromycin. We have seen that it binds to the 50S ribosomal subunit and it prevents the translocation of the ribosome. Thereby, it prevents the protein biosynthesis pathway. Here we can see that they are active against gram-positive bacteria, Chlamydia and Rickettsia. They are also useful in treatment of pneumonia. We have also seen they are useful in treating organisms which show resistance against penicillins. There are certain drawbacks, as use of macrolides causes certain eyes infection in kids, we have also seen that their use may cause some gastrointestinal problems. We have seen that this drug causes some adverse effects on certain organisms like Enterobacteriaceae. They are normally showing resistance against macrolides. The reason behind is their outer wall actually prevents the entry of the drug. Lincosamides comprises of lincomycin, which is produced by Streptomyces lincolensis, and it also has got a semisynthetic derivative called clindamycin. They are active against gram-positive as well as gram-negative bacteria, specifically aerobic gram-positive cocci and anaerobic gram-negative bacilli are the target sites of lincosamides. They are specifically used in ointments to treat acne. Streptogramins actually comprises of two different compounds called group A streptogramin and group B streptogramin. Group A streptogramin comprises of a 23-member unsaturated ring comprising of lactone and peptides. We do have another group called streptogramin B, which is a lactone cyclized peptide. These two are of different structurally, but individually they are executing bacteriostatic function, and when they are used in combinations, then they show bacteriolysis or bactericidal action. We will see that the streptogramin A or streptogramin B, both of them target the 23S ribosomal subunit of 50S ribosomal subunit. Here we will see that they prevent the peptidyl transferase activity. First, the binding of the streptogramin A actually makes the conformation of the ribosome suitable for the binding of streptogramin B. That's why we have seen that when these two drugs are used in combinations or in synergy, they show better results. Now we will be discussing about oxazolidinones. This group of drugs were discovered after the discovery of streptogramins in 1962. We got this drug after 2000. These class of drugs were specifically useful in treating S. aureus infections wherein they show resistant against beta-lactam drugs. So these drugs are used to treat or combat diseases which are MDR, means multiple drug resistant strains, especially vancomycin resistant bacteria were susceptible to be killed by oxazolidinones. The most important drug of this class is called linezolid. Here we will see that these drugs actually execute their action by preventing the association between the 30S and 50S ribosomal subunit. Thereby they have inhibited the protein biosynthesis pathway. With this actually we can summarize the protein biosynthesis inhibitors. Therein we have seen a class of drugs that are targeting 30S ribosomal subunit, some of them are targeting the 50S ribosomal subunit, and finally we have seen that the oxazolidinones are actually restricting the association between the 30S and 50S ribosomal subunit. Antimetabolites were supposed to be the first antibacterial drugs that were discovered in 1930 by Gerhard Domag. Basically, what are antimetabolites? Antimetabolites are chemical compounds that mimic the various metabolites which are synthesized by the bacteria during its existence or during its life cycle. So antimetabolites were utilized to mimic the action of the metabolites, and what they did, they actually occupied the active sites of the enzymes, thereby they restricted the action of the enzymes by executing competitive inhibition. the most important antimetabolite that was used was sulfa drugs or sulfonamides. Actually, the bacterial system has to synthesize its folic acid on its own. It doesn't procure folic acid from the environment. That's why it will synthesize folic acid utilizing certain precursor molecules. Among one of them is para-aminobenzoic acid. Sulfanilamide or sulfamethoxazole structurally mimics para-aminobenzoic acid, and by its structural similarity it binds to the active site of the enzyme which is involved in conversion of PABA to the next metabolite. So thereby it restricts the conversion of PABA and finally it prevents the synthesis of folic acid. So this is the mode of action of sulfamethoxazole. So the benefit of utilizing this sulfamethoxazole is that our system, we do procure folic acid through our diet. So we don't have the enzymes which are needed for the synthesis of folic acid. This is how we have attained selective toxicity against this sulfonamides. These drugs are useful for treating various chlamydial infections and shigellosis. They are used to treat urinary tract infections, but there were again certain drawbacks or limitations, as this may cause aplastic anemia, this may cause depletion of blood cells, this may cause various side effects.
Ma'am, I have a question. Which one between sulfa drugs and protein biosynthesis inhibitors has higher therapeutic index?
Actually, sulfa drugs have got sufficiently high chemotherapeutic index. The reason behind is quite logical, that folic acid biosynthesis pathway is absolutely present in prokaryotes. Eukaryotic system don't have this folic acid biosynthesis pathway, as they uptake the folic acid through their nutrients, as we were discussing. This is the reason that we can have a sufficiently good chemotherapeutic index in case of antimetabolites or sulfa drugs, whereas in case of protein biosynthesis inhibition, as I have said, that there are certain mitochondrial genes which get transcribed and translated, which utilize the 70th ribosomal subunit, thereby the chemotherapeutic index falls off a bit. These drugs are specifically useful in treating infections, urinary tract infections, shigellosis, and they are useful in treating infections in the eyes and burns, wherein they are added in the ointments and solutions which are used to treat eye infections. The major drawback in case of sulfa drugs is that various organisms have developed methods to uptake folic acid from the environment. So they are not solely depending on the folic acid biosynthesis pathway, which is the site of action of sulfa drugs. The second way of developing resistance against the sulfa drugs is that they are procuring certain plasmids, and these plasmids are coding certain enzymes. These enzymes have got very low affinity towards these antimetabolites. So we are adding these antimetabolites, but these antimetabolites are not being binding to the enzyme's active site. Another antimetabolite drug called Trimethoprim is used to inhibit the folic acid biosynthesis pathway. It targets the second step of folic acid biosynthesis pathway, wherein it interacts with dihydrofolate reductase and prevents the conversion of dihydrofolate to tetrahydrofolate. This is the way how trimethoprim executes its effect on preventing folic acid biosynthesis. We have seen that trimethoprim is used for the treatment of urinary tract infections. It has been used in the treatment of respiratory tract infection or in infections in the middle ear. In these cases, we use trimethoprim. But there are also certain drawbacks which has led to the use of combination of these two drugs, trimethoprim and sulfonamides, together. nowadays the combinatorial therapy has become very much effective. The reason behind is, if we use sulfonamide and trimethoprim together, then what happens that we are targeting both the steps of folic acid biosynthesis pathway. So if some bacteria have developed some means of restricting or inhibiting one of the pathways, then obviously the second pathway is there, or the second enzyme is there to inhibit the folic acid biosynthesis. So the net result is that if these two antimetabolites join hands together, then the result will be much more effective and we can combat the pathogen in a better way. In all, there are 5 different classes of drugs that are used to combat Mycobacterium tuberculosis infections, namely rifampicin, streptomycin, isoniazid, ethambutol and pyrazinamide. Isoniazid basically prevents the mycolic acid biosynthesis pathway. It is an antimetabolite that mimics certain vitamins which are the building blocks of mycolic acid. Next we have got ethambutol, which is a drug that inhibits the important pathways which are undergoing inside the mycobacterial system. It is also executing a effect which is not yet known, that how it executes its inhibitory action. So these drugs are used in different combinations. There are certain cases wherein we see that isoniazid is ineffective. Under such circumstances we use ethambutol. So mycobacteria is a tough organism to combat. That's why combinatorial therapy in different combinations are used to combat mycobacterial infections. In this lecture, we have discussed about protein biosynthesis inhibitors. We have also discussed the role of different antimetabolites in restricting the growth of bacteria, and finally we discussed about the various antibiotics that are used to combat Mycobacterium tuberculosis.